afternoon, everybody. My name is Dr. Kate Delaporte. I am the curator of the Waite Arboretum and the Waite Conservation Reserve. I am coming to you from the Coach House, the Waite Research Institute at the Waite campus and the University of Adelaide, South Australia. Today, I'm going to share with you some of our findings about regional impacts on Leptospermum growth, bioactive honey plantations in South Australia. This work is part of our project, which is called Leptospermum for Bioactive Honey Production in South Australia. We are looking to support a bioactive leptospermum breeding program for South Australia. We were doing this through 3 ways. The first one was looking at genetic diversity of superior lines. We're looking at propagation methods, and we're looking at the influences of environment on plant growth, flowering, and nectar, including DHA production and sugar production. This project, or this talk today, is specifically going to focus on the environmental influences and how we've looked at gathering that information. This talk today is going to cover a bit of background. Why are we doing this? Where are we doing it? What are we doing? And what did we find out? Firstly, what is bioactive honey? Probably all quite familiar with bioactive honey. It's a honey that contains bioactive compounds. Bioactive compounds are phytochemicals that provide a suite of biological effects. They have antibacterial, anti-inflammatory, antioxidants, and they may also support the immune system, which is really great for human health. Bioactive honey can be sourced from the nectar produced by a number of different plant species in Australia, including plants from the genus Leptospermum. Leptospermum honey can be known as Manuka honey, and it is currently in hot demand around the world. Research has identified a specific set of phytochemicals within Manuka honey that are contributing to the bioactivity, the most important being methyl glyoxal. Recently, there has been renewed interest in Leptospermum for the production of bioactive honey. In 2015, Australian researchers began a comprehensive study of all of the Leptospermum species in Australia to determine which ones could produce bioactive honey, and from there, this project was born. Our questions are very much focused on the plant by environment part of the process. Which species might be best suited to plantation production of high-grade medicinal bioactive honey in South Australia? Always influencing our thoughts. Manuka equals Leptospermum scoparium. People want to plant scoparium to produce bioactive Manuka honey, but is that the best species for South Australian production? Across Australia, we have species with really high levels of DHA in the nectar that grow in a wide range of environments that can be hybridized to produce attractive and adaptable plants that also have bioactive nectar. Should we be looking at those? In South Australia, we have 5 endemic species of Leptospermum and 1 introduced species. Research has identified that the 3 species exhibit the potential for bioactive honey, and 2 of them are possibly better than scoparium. The 2 highest DHA yielding species are Leptospermum continentale and Leptospermum lanigerum. Would our local species do better under local conditions and be more likely to survive with lower input compared to scoparium? The State Flora Catalogue for South Australia shows which Leptospermum species they provide. A quick review of the availability: scoparium is sold mainly as an ornamental plant for gardens, but this catalogue gives us good information on the growing conditions that it needs, and this was used as a resource to help guide our thoughts. For our project, we looked for field sites where plants were growing different species of Leptospermum in different environments. Early on, we identified that water was arguably the key limiting factor in growing Leptospermum scapparium as the primary species for honey production, but we needed to determine if it was true. Back in 2015, I did a quick and dirty comparison of where Leptospermum scapparium occurs naturally and what areas of South Australia might be suitable to grow it. Rainfall is not just about annual totals, but also about distribution. While lots of areas in South Australia have adequate rainfall, the distribution differs a lot from the Tasmanian and Victorian sites where scapparium grows naturally, such as Burnie in Tasmania, Swansea in Tasmania, Warrnambool in Victoria, which is the darker grey. Compare that to Wiradjuri Forest, the dark brown line. Mount Gambier with the high rainfall is the grey line. You can see from the lines here that in general, our peaks and troughs of rainfall are different from those where scapparium occurs naturally, particularly the troughs. How much of an influence would this have on plant survival and nectar production? Generally, our local species are well adapted to different soils and soil types and pH levels and are moderately tolerant to frosts. But the key thing is the rainfall. The endemic locations of the species tend towards the 500 mil plus for rainfall. In South Australia, we also have a Mediterranean climate, which sees most of our rainfall come in a blob between autumn and spring, and we can have long periods of zero rainfall with high temperatures and high evaporation, which rapidly lowers the subsoil moisture levels. Plants that thrive here must be able to cope with having a dry root zone. Not sure how scapparium was going to cope with that. In order to find out as much as we could in the short time frame of the CRC, we located field sites that had already been established. We originally had a field site at Keith in the southeast, which was borderline to scapparium, and despite a wet winter for establishment, the following dry summer wiped out the entire plantation. All of these sites we've used are irrigated with the exception of Macclesfield. 4 of our sites, Charleston, Mylor, Bridgewater and Macclesfield, are located within a 30-minute drive of the Waite campus. They are in the Adelaide Hills and it rains a lot. Port Vincent is on the Yorke Peninsula and Wiradjuri is in the mid-north of the state, a good 3.5-hour drive from Adelaide. All of these sites were growing Leptospermum species for honey production. We're going to present site-specific data for 3 sites in particular, Wiradjuri, Port Vincent and Bridgewater. These 3 environments differ in rainfall, temperature and soil type and provide data on the areas most likely to be used for growing Leptospermum. Bridgewater represents the Adelaide Hills, being Mylor, Charleston and Macclesfield, and is naturally high in rainfall. Wiradjuri is right on the recommended rainfall for scapparium and Port Vincent is quite dry. Going to drill down into the site specifics here, and the first data is rainfall. As you can see, the rainfall from each site was variable over the 3 years of the trial, with 2 of the 3 years being well below the 10-year average, and 2019 was the driest year of our trial. You will note that Bridgewater is the wettest of our 3 sites, averaging 1,000 mils per year. Wiradjuri is next with about 500, and Port Vincent has 380 as its 10-year average. Let's look at site-specific data. Excuse me. Over the 3 years of data collection at Bridgewater, rainfall was consistently lower than the 10 and 50-year averages, particularly in the autumn and spring periods. And we also had this really strange lack of rain in July. This has the effect of contracting the main growing period and extending the period with little rainfall. And this has reinforced why supplementary irrigation is so important for establishment and growing of scapparium. From the sites we looked at, the mean, maximum and minimum monthly temperature data is shown on each slide. However, the recorded temperatures didn't differ greatly from the long-term averages, and so I'm not going to be discussing them at length, but it may come out further in our deeper analysis that temperature data is having an effect. This is Warabra. Same pattern was observed, with the exception of these randomly high falls in February, April, and October last year. Across the 3 years of the program, the rainfall was consistently lower, and we again had this lack of rain in July, dry autumn, and then suddenly a quite dry spring. Irrigation is so important. Port Vincent, our next site, similar pattern, low rainfall, July was low, low spring, and again across the whole site below average. The 5-year mean is starting to dip below the longer term trends, as we might expect from climate change. Again, temperature data is not really seeming to tell us much at this stage. So apart from looking at the environment that was influencing how the plants might grow, we actually also did some field observations and measurements. At each of the 5 sites, we tagged 20 individual plants, and from each plant we made observations and took height and width measurements every year. Nectar was also collected from each plant. Our Warabra site was reviewed every 3 months, but the other sites were visited only once a year. Looking at the height and width growth measurements, we can see an interesting pattern. The plants at Port Vincent and Bridgewater, which are our first 4 columns, here and here, were already quite well established when we started the trial. And so they have actually grown very little in height over the time of the trial, but they have quite well expanded in their width. The plants at Warabra, these plants here are seedlings when they were planted, so they were 15 centimetres tall, and they've grown as expected to 130 centimetres tall in the 4 years that we've measured them. And they have also widened quite considerably from very, very narrow plants to being now 140 centimetres wide. The other plants from Warabra were established from cutting grown plants that were already 2 years old, so their growth has been quite regular, but not a great deal of growth. Once again, they have reached their maximum height or close to a maximum height because they've started flowering, and we can see that their width has thickened up as well. Once the plants settle into regular flowering, their upright growth almost stops and they just become bushier. And this is evidenced by the small changes in height, particularly with the Bridgewater plants. All the plants across all sites are now flowering consistently, and we have started at the 2021 data collection at Warabra, and we'll be looking to do the final data collection at the other sites before the end of the year in the next flowering period. We collected nectar from plants at each site. So we collected nectar from 20 flowers from each of the 20 plants at each site to have a duplicate sample, which was analysed for total sugars, DHA ratio, and the estimated sugar mass at the University of Sunshine Coast. So I'm sharing here with you some results. The average reading for each year from the 20 plants is presented in the average column, and the range, which is the real minimum value recorded and the real maximum value recorded, is presented here as the range. So as you can see, we've got quite a degree of variation between sites across years, and there's quite significant difference in the range. And it does depend on which species we're looking at. So even Scoparium at the same site is quite different across years. The estimated sugar mass average has the same level of variability. So you can see this variability is a bit of a concern, and we really do need at least another year of data to try and get to the bottom of what this variation is being caused by. Is it genetic? Is it environmental? Or is it method driven? Are we actually collecting and analysing this nectar in not the best way? So along with our measured data, We made general observations on how the plants were growing at the different sites. The key theme that we determined was that each site obviously is different, and a lot of how those plants grow and respond to those sites depends on the origin of the plant material and the variability of the soils across the site. Most of our sites, the Scoparium plants, came from southwestern Victoria, which has a climate as close to the climate of South Australia out of all the sites where you could possibly get Scoparium from. Polygalifolium, which we had at 2 different sites, comes from New South Wales, Queensland. So how they're going to respond under South Australian conditions is very much unknown. Flowering was dependent on where the original plant material came from. It didn't seem to change with location. And yes, of course, you can overwater and underwater, and the water quality is very important. All of these things can negatively impact plant growth. Plant maintenance is an important part of establishing a plantation, and general observation. Very similar observations to what you would expect for any woody perennial plantings. You need good weed control, regular watering, minimise herbivore grazing activity, and invertebrate pests also need to be considered. We did not find any issues with nutrients, and no fertilisers had been added at any site. Plant spacing is a hot topic in our tea room, but really it's a personal preference and depends on your site and what you are trying to achieve. Whether it is close spacing to form a hedge with higher early production of flowers, or wider spacing to minimise inputs early, nothing really seems to make that much difference to how the plants are going to grow. It's too early to tell. We are left still with some ongoing questions. For our big question, how does the environment affect plant growth? We've got some idea. Obviously, plants enjoy regular watering and cooler temperatures. That's a given. They are going to grow better with regular water, and if it doesn't get too hot, all the better. The young plants will grow fast, and once they form a suitable scaffold, they'll body out, they'll thicken out, so provide more opportunities for flowering tissue. Flowering time seems to be dictated by origin rather than the season or the temperature, but we do not have enough data on that to be really adamant about that yet. So my other ongoing questions, and we may be able to bring information to the table in the final 6 months of the project, are looking at all about water and nectar flow. Now that we have consistent flowering across sites, we can look more deeply at flowering and the nectar flow and the influences that might be happening. So this has been a very quick review of the work we've done through this project in the CRC, with lots of time spent out in the field. It's been great. We've had some concluding points to take home. The most important, we think, is that you really need to work out what you want to produce from the plantation, both honey-wise and financially, with a focus your planning and plant selection around that. And of course, don't forget about the bees. This work has been financed by the CRC for Honeybee Products, Project 10. I'd like to thank the CRC for enabling us to do this project. I would like to thank Nick for all of his work, and to Phil Ashby and Kev and Annette Webb for their roles in this project. Nick was supported by the CRC, I'm supported by the University of Adelaide. Nectar analysis was provided by the University of Sunshine Coast and supported by the CRC for Honeybee Products. Thank you very much.